and we also have a number of blooms in other systems, which I will show. I also get to use some ecological theory and tools to help understand why these blooms occur and why they produce toxins. So just to give you an idea of where we're going to go today, first I'll just give you a background on what is this stuff on my backgrounds, and a little bit of ecological context to what it means. And then of course I'll say, well, what favors these sorts of phytoplankton, and of course my title, my talk title, and what you see there is that nutrient ratios matter. There's also other things that matter that we'll talk about as well. But then I'll go through, just as Ben said, I'll talk about some empirical studies that were done, and then some large-scale tests to see exactly if those are correlation or causation, and then I'll sum up. So first thing to know is that there's a lot of algae out there. There's a lot of diversity, and there are 6 major groups, and Cyanobacteria just are one of them. They're the Cyanophyta group. So there are all kinds of species of algae that are there. They live in just about any water, in crust of desert soil, just about all over on Earth, even in sloth's fur, these algae live. So what about these cyanobacteria is making them harmful? So first off, the harmful algal blooms, or HABs, maybe you've heard that term, in Kansas, are made up of this blue-green algae. Technically they are prokaryotes, but they function and they look just like algae. And so the issue with these blooms is that they can produce really potent toxins, even more potent than cobra venom, some of the toxins that they produce. They can cause taste and odor issues in drinking water supplies, but not always, and we don't know why. We don't have a good grasp on that. Within cyanobacteria, there are many different forms. So there's somewhere between 2,000 and 8,000 species. We know of about 2,500 today. Each species has hundreds or thousands of different genetic strains. And only some of the strains can produce the toxins and the taste and odor metabolites that I just referred to. And they have to have specific genes to produce that, and not every strain has that gene. Additionally, not every strain looks the same. There are many different morphologies. So here we can see one that's sort of, these are individual cells in a long filament. We can see these are straightened filaments that sort of bind together. And then you can even see, here's a single cell that they come in these large colonies. So they kind of group together all these cells in this goo, which is exactly what you see in the background. And these 3 species bloom in our reservoirs here and throughout the world. Another key thing that cyanobacteria do is that they have these cells called heterocysts. So these are these clear cells that you see here and here. And those heterocysts allow them to fix atmospheric nitrogen. So just like there are microbes intertwined with soybean roots that allow them to fix nitrogen, legumes, cyanobacteria have that same ability to take in nitrogen from the atmosphere and use it for growth. Of course, above all, these blooms are gross. And unfortunately, they are happening throughout our state in many different locations. So here is a graphic from KDHE's response program, and you can see where some reservoirs have been closed, or watches or warnings have been posted because of these blooms, and they're at numerous locations, including Milford and Marion and Webster and other large reservoirs in the state. To most people, they're aesthetically unpleasing. They decrease property values, but they also disrupt food webs. So here are the herbivores, the Daphnia, that can eat these blooms. And what you can see here in the background is these are some of those straightened filaments that I showed, and also these curly filaments, and they are really large compared to the Daphnia that need to eat them. They're hard to eat. It's sort of like me giving you a 2x4. It takes a long time to eat. Maybe you can eat it, and it also turns out that the cyanobacteria are not very nutritious, just like a 2x4. So it's hard. They kind of clog up the system. And of course, they produce this suite of potent toxins and taste and odor metabolites. So let's go down that path just a little bit. The toxin metabolites that they can produce, there's a wide suite of them. Some of them are hepatotoxins, which means they affect and shut down livers. Some of them are neurological toxins, and some of them are dermatoxins. They create rashes on your skin. The most common is a toxin called microcystin, and it actually has a number of different forms, over 200, or congeners, and they range in toxicity. And just put this in your pocket for later, they also vary in their nutrient proportions. So here you can see microcystin LR, which has these 2 amino acids, leucine and arginine. You can tell between those two amino acids, they have different amounts of nitrogen, carbon, and oxygen in them. And so this LR could be RR, LY, LR. There are all sorts of other versions of this. And so we will talk more about them at the very end of the talk, but just know that there are a wide range of toxins that they can produce. And of course, unfortunately, they are linked to illness and death of animals and humans. Human fatalities are rare, but they have occurred. So they do have some serious consequences associated with them. They also produce these earthy, musty, fishy, swampy smells and tastes in your drinking water. One of the compounds that cyanobacteria produce is geosmin. It's the smell after rain, that dirt smell, but cyanobacteria are one of the primary causes of that smell, at least in freshwater systems. They also produce other compounds like 2-methyl-isoborneol or 2-MIB, which again, they create these taste and odor issues in finished drinking water supplies. Just to note, you'll notice there's no nitrogen in these. They're both carbon rich. Again, just put that in your pocket for later, but these only have carbon, oxygen, and hydrogen in them. They're really difficult to remove for the drinking water plant. So that water comes from a reservoir. Let's say here in town, we have Clinton Lake. A drinking water plant tries to treat it. And these taste and odor compounds, we can sense them at 5 parts per trillion. So they're very difficult to remove. And of course, although they are not harmful in any way to our knowledge, when you get water that smells like dead fish or grass, you still have a little bit of decrease in your confidence that you're getting clean water. Cyanobacteria also have the specialty, and exactly what I'm showing in the background of my slides. These surface scums. These can form out on a lake or on a tank, which I'll show pictures of, but they also pile up in bays. So they grow throughout the water column, throughout the lake. Then you have a calm period where they have these buoyant little balloons in them, allows them all to get to the surface, and then wind blows them into one corner. And that is what you're seeing here on Milford in the military marina. These scums also, because they're buoyant, they can be moved around. They can change orders of magnitude in one spot, just over several hours because they move, they're advective. So they can rapidly change the location of the lake, and they are the major health hazard because they represent an accumulation of lots of cells. And we actually have some data on this. This is from a Dutch report. What you can see here is essentially the toxin. Again, this is the toxin out in the lake. Those are the blue. And you see the toxin in the scum that piles up along the shore here in the purple. And then here is the toxin. This red line, this is time. This is in a few Dutch lakes. This is the line where if you surpass that, you start seeing effects from the toxin, some negative effects. And what you can see is the scums always are above that line. They aren't always 100% above the line in our lakes, but again, that is the major health hazard here. So why? Why do these things occur? Why do blooms occur in general? And so it's important to remember that algae are just tiny plants, and plants need really basic stuff to grow. They need light, they need water, and they need nutrients like nitrogen and phosphorus and potassium. Each group of algae has different adaptations. And so this poses the question, well, what favors the gross goo that you've been seeing? What favors the cyanobacteria? There are 2 primary factors that get reported over and over again. One is climate change, or in this case, blooms like it hot for a catchy tidal. Here you can see on the y-axis, we have the percent of maximum growth rate, with 100 being where you grow the best. And you have that versus temperature. You have 4 of those 6 major groups that I showed at the beginning. And what this shows is when it gets hot over 25 or 30 degrees centigrade, there's really only 2 groups, cyanobacteria and these green algae or chlorophytes, that can compete. So temperature alone can help favor the cyanobacteria, but nutrients do as well. And they do really well when there's a lot of nutrients. So here we have 2 y-axes, although they both range from 0 to 1 or 0 to 100%. One is how much of the fraction that cyanobacteria make in terms of lakes. So this was a study over a wide number of lakes in Iowa. And here's the risk of a bloom within this wide number of lakes. I believe it was 30 or 40 lakes. So here we can see total phosphorus increasing and total nitrogen increasing. And what you see is that the risk of having a bloom and cyanobacteria making up a lot of that algal community increase as nitrogen and phosphorus increase. So those are the 2 main factors. But then how much nitrogen and how much phosphorus is there also matters, and the proportion of them also matters. So here's a study from 1983 by Val Smith. He used to be in KUEEB. And he was the first one to really show this, that here you have the blue-green algae or the cyanobacteria as a percentage of the algal community. Here you have this total nitrogen to total phosphorus ratio, or nitrogen to phosphorus ratio. Each one of these letters represent lakes from around the world. And so he was the first one to group this together and show, well, something here, they don't have to dominate when this N to P ratio is low or less than 30:1, but they can, and they really can't dominate when that N to P ratio gets higher. So what this is saying, and we'll walk through this in a second, is it begs the question, well, does that mean that cyanobacteria are really good competitors for nitrogen, right? Because they are down here where there's not much nitrogen compared to the amount of phosphorus. But when there's a lot of nitrogen compared to the amount of phosphorus, they appear to be bad competitors. Does that mean they're bad at phosphorus competition? What does this mean? Is this real or not? So first, let me walk you through why it might be real and not just a simple correlation. So here we have the nitrogen to phosphorus ratio drawn out by how many elements. And if you're the average algal cell, then you need 7 nitrogens for every 1 phosphorus that you take up. It's called the Redfield ratio. We will not get into that today, but just know that you're the average algal cell and you need 7 nitrogens for every 1 phosphorus. So if you add a lot of phosphorus, in this case, nitrogen becomes limiting or deficient, right? Because you gulp up your 7 nitrogens, you gulp up 1 phosphorus, but then you don't have any more nitrogen that you can eat, that you can assimilate into your cell. But there's plenty of phosphorus here that can be used. So one way cyanobacteria might do well when there is not a lot of nitrogen in the system is they can fix it. Remember, some of them have the ability to fix nitrogen from the atmosphere. So if you don't have enough, well, you can just make it. You can just pull it straight from the atmosphere. And that's what these gray nitrogens represent, so that you can finally use the rest of this phosphorus. Alternatively, if you were to add a lot of nitrogen and not have enough phosphorus, then that would be the reverse, where phosphorus would become deficient or limiting, because you can use all these nitrogens and all these phosphorus, the phosphorus get used up, there's still more nitrogen that can be used. So now the question is, well, was Val's original graph, was that a causation, or maybe it was only correlation? And the only way to find that out is to run tests. And so I'm going to show you some of those tests that we've run. So during my master's, I went out to the University of Idaho, and there was a lake in the desert in Oregon that frequently got these blooms. So it looks very nice now. It's catchment. There's the Blue Mountains in the background. And they impounded this lake because of flood risks. But unfortunately, for most of the summer, it looks exactly like this. It's not this blue paradise and oasis in the desert. It is this gross, scummy, cyanobacteria-rich lake. So we said, if we know that this happens every year, let's run some experiments. So my master's advisor and myself, Frank Wilhelm, we said, okay, let's manipulate the nitrogen to phosphorus ratio by adding a lot of nitrogen. And if cyanobacteria really can't compete in this high nitrogen to phosphorus ratio environment, then their population should decrease. So we built the biggest enclosures that we could, which I'll show in a second, about 70,000 liters. And the goal was to just increase this ratio well above that 30:1, way above that, over 100. And so we added ammonium nitrate to do that. Our enclosures that we used, well, they were very easy to draw up. So here we had 3 enclosures. We used the lake as a control because the enclosures were so large and difficult to make. And that reservoir out there has a process called stratification, which essentially separates the water column. And so what happens is you get warm water up near the surface and cold water down near the bottom. They don't mix. That happens in the enclosures too. So it seals off this top area to do an experiment. They were open at the bottom to allow for zooplankton migration and sort of hung taut, and they were hung from a dock. So this was a really nice drawing, but then putting it into practice, it's a little bit bigger. So this is a person right here, just to show off some of the scale. And here's the enclosure laid out, and we're putting tubing around so that we keep that up. Here's what that enclosure looked like in the lake. And then here's just a view kind of backing out, right, where you can see the dam and you can see these large enclosures, but they're not that large compared to the reservoir. So we went out, we put these in June. So here, this is date at the bottom, June, July, August, September, October. The lake will be in green. So for now, in the slides coming up, and the enclosures will be in blue. And here's your TN-TP ratio, or nitrogen to phosphorus ratio. Our goal was to get it over 100. And you can see my shoddy master's student, intro master's student math definitely got it over 100. And so I definitely missed just a little bit on that because the enclosures move and sort of come in and out, but we definitely accomplished our goal of getting it over 100. So we added nitrogen here, and then we sort of let that fall, and we said, well, we want to keep it over 100, so we added a little bit more nitrogen. That's what this represents here, right at the end of August. But then we sort of just let everything happen and let that ratio fall. So you can say they start and end in the same place, but obviously the enclosures have much different condition. So what about the algal community? Well, the lake, like it is almost every summer, if not every summer for the past 20 years, was dominated by cyanobacteria, and particularly those that could be toxic. Again, doesn't mean that they absolutely produce the toxin, but they seemed to have the ability. So here you have time, and here you have the biovolume, or the percentage, right? So cyanobacteria just gradually just take over and represent almost 95% of all algae in the lake. So what happened in our enclosures where we added all this nitrogen? Well, weirdly, at first, it spurred a little bit of a cyanobacteria bloom, but that was really short-lived. And after that, this big green blob here represents green algae, or those chlorophytes, the ones that are also good at high temperatures. And they dominated that community. And cyanobacteria, yes, they were still there, but they represented a small portion of that community. So now the question is, well, what happened to the toxin? So here you've seen this graph before, and what I'm going to do is overlay the toxin on top of this. So here now I've overlaid the toxin on top of this. Again, green is the lake. So the lake went toxic, right? Was not toxic until about mid-August, and then there is some toxicity there. It is present. You can see it goes above the limit of detection. It reaches concentrations which are somewhat harmful, not to that moderate level, but yes. And what you see is the enclosures. Well, right when we sampled that first nitrogen addition, that there were cyanobacteria there, and they were producing quite a lot of toxins, but that quickly was eliminated because there's no cyanobacteria to produce them until this nitrogen to phosphorus ratio went below about 30 or 50 to 1. Sometime in there, cyanobacteria started to produce toxins, and then they ramped up very quickly towards the end of the season, even though in the lake, you can see the toxin is going down, but in the enclosures, it went up. So sort of a summary of what this experiment was about is that we did increase the N/P ratio a lot, and it did indeed exclude most cyanobacteria, not all, but most. It also eliminated the toxins that are associated. They're not there. If there's no species to produce the toxin, then there is no toxin. So that is why there wasn't toxin there. But once cyanobacteria were favored, that N/P ratio came lower. As you saw, toxin production then really ramped up. And so one thought was this was, well, if we added all this nitrogen, what cyanobacteria are favored, and they have this nitrogen-rich metabolite in microsystem, that maybe they have to deal with all this extra nitrogen. Well, maybe they produce more nitrogen-rich metabolites, some way to shunt that, either that go in the cell or outside of the cell to get rid of all this backup of nitrogen. And of course, overall, it supported these N/P ratio effects that were observed empirically. So we found the same thing that we're finding empirically. So that leads towards that it is caused by N/P ratios, or at least in part. But that gets us onto a different question that we took away from that. Well, what about substitutable resources? And substitutable resources are sort of like nitrate and ammonium. These are both nitrogen forms. If you're an algal cell, you can take up either one of these. You have the ability, but does that matter at all? Do those proportions matter at all? Non-substitutable are things like nitrogen and phosphorus and carbon. No matter how much nitrogen you have, it doesn't make up the deficit that you have in phosphorus, or vice versa. So the question was, okay, well, what about different forms? And do these non-substitutable and substitutable resources interact to help drive community composition? So we again looked empirically. We said we already have some data on this. So we looked at Cheney Reservoir, which is the drinking water supply reservoir for the city of Wichita. And we looked at Eagle, Morris, and Geist reservoirs around Indianapolis, which are their drinking water reservoirs. Because they're drinking water reservoirs, they have data on these taste and odor compounds as well. So they have the toxin, but they also have data on the taste and odor compounds. So first, let's look for trends with the N to P ratios. And so of course, this is just a temporal graph, but what we can see here is Cyanobacteria biovolume on the Y. Here you can see the nitrogen to phosphorus ratio, which is this red dashed line. We also have what percentage of the community had the potential to fix nitrogen, that's grayed out. And then the parts not grayed out, that's Cyanobacteria that cannot fix nitrogen. And then here we have time, this is in years, so that's 2001 to 2013. And you'll see a series of graphs like this. So here's Gini, and what you can see is when this red line goes very high, there isn't much Cyanobacteria, especially with nitrogen fixers. Here in 2006, maybe it spurred a little bloom, but not nitrogen fixers. So we see the same pattern that Val saw originally in his data. When we look at Eagle Creek, Geist, and Morse, we see that same thing. Again, there are times where you see an increase in the nitrogen to phosphorus ratio, maybe here, and you also see an increase in Cyanobacteria, but in most situations, what you can see is the ratio starts high, it goes low, and then Cyanobacteria increase. Sort of just like what we see in Willow Creek. So now let's lay out some graphics where we conform this ratio, but also think about what nitrogen and phosphorus do independently. So in this situation, we have nitrogen on the Y, total phosphorus on the X, and increasing. This line represents 30 to 1. So there's 30 nitrogen to 1 phosphorus along this line. So anything below this line, you can kind of think of it as nitrogen limited. Anything above, you can think of it as phosphorus limited in this version of the graph. And here I've just color-coded points of the toxin microsystem. So when we see that toxin, we mostly see it just as we did in our enclosures, and as others have found, when the nitrogen to phosphorus ratio was low or below 30 to 1, where Cyanobacteria are favored, we see their toxins. Makes sense. What about these carbon-rich metabolites, though? Where do we find those? Well, that's a little bit more complicated of a story. There are other organisms and other bacteria that can produce geosmin and MIB. So here we have geosmin again. Anything above 5 nanograms per liter, we can pick up, we can start to smell, especially above 10. All humans can smell it then, this sort of musty, the smell after rain. And you can see that in the water, there was a handful of positive samples, but that, again, when we have the highest concentrations, it seems to be at a low nitrogen to phosphorus ratio. That makes sense. Again, we have the same graph with MIB. You can see the most potent was actually a little bit higher N to P ratio than 30 to 1, but the majority of the points still hover around this bottom portion of the graph where the N to P ratios are low. So not infallible, but again, there's some interference there. There's some noise. So great, we kind of found the same thing that of course Val originally found. Jennifer Graham, who used to be here, Diane Orihill found the same thing in Canada, and of course myself and team did in 2014. But we really need a better way to produce these predict these metabolite producing blooms. And there's this hypothesis that Cyanobacteria are better at these chemically reduced, meaning ammonia, nitrogen forms compared to the oxidized forms. So we wanted to know, well, do the proportions of nitrate and ammonia, these substitutable nutrients, do they matter? Is there anything to do with these proportions? And when it's more reduced, the low, so that way the ammonia means that it has more proportion compared to the oxidized form, does that have any effect on the community? So same graphs as the N to P ratio, now we have the nitrate and ammonia ratio over here. You can see there's a wide range. And again, you can see when the red line is sort of peaks, the Cyanobacteria do not. You can kind of see that over and over and over again, that there are not Cyanobacteria when the nitrate to ammonia ratio is high. In this case, we deemed that over 5, which you'll see in a second. So here, again, the nitrate to ammonia ratio over here, notice that every system is different in terms of the range of nitrate to ammonia ratios. But again, we see the same thing. When the nitrate to ammonia ratio is high, meaning there's a lot of nitrate and just a little bit of ammonia, Cyanobacteria are low, and vice versa. Same graphs we had before, but instead of total nitrogen and total phosphorus, we now have nitrate and ammonium. And note that when it comes to this toxin, this is the toxin again, we see that all these points of the highest toxins happen when there's not much of either, but there is a little bit more of ammonium. So at low nitrate to ammonium ratios. So that ammonium is making up the biggest part that reduced N. What about for geosmin? We sort of find the same thing, that it's down here when there's not very much nitrogen in general, maybe favoring nitrogen fixers. But also, most of the points are on this side of the line. In this case, this is a ratio of 5:1. That's what this line represents here. So 5 parts nitrate, 1 part ammonia. So again, they're all on the side where it is ammonia rich. MIB, again, more of a scattering all over the place. But again, you see a number of samples down here where ammonia is the dominant nitrogen form there. Okay, so the ultimate question. Well, are nitrate and ammonia, or this nitrate to ammonia ratio, are they correlated with unwanted metabolites from these different sorts of cyanobacteria? So what we did is we parted those out. We saw that in the data set, when a cyanobacteria bloom happened, at least in that data, it was either dominated by nitrogen fixers or dominated by non-fixers. There was a little bit of intermingling, but what we did is when it was dominated by fixers, we divided how much metabolite there was by the biovolume of fixers, these nitrogen fixers, right? And we did the same thing for taxa that could not fix nitrogen. This is a PCA plot, a principal components plot. Really, the only thing you need to know here is that groups that are tightly grouped together are more correlated. Groups that are opposite of one another are inversely correlated. And the separate groups means that they are correlated with just what's in their group and not correlated in another group. So the closer you are, the more correlated you are. In here, so you can see the metabolite per biovolume for MIB, microcystin, and geosmin. Fixers here, they all group together. And the non-fixers all group together. But they also group with 2 of the nutrients, total phosphorus and ammonia. So they group with this reduced nitrogen form. Total nitrogen here, you can see, is almost opposing total phosphorus, right? So they're inversely related, and nitrate was highly correlated with total nitrogen. So the next slide shouldn't be that surprising, because we can see these same groups, again, the fixers group together and the non-fixers group together. And now I've put it against the ratio. So this is the nitrate to ammonia ratio and the TNTP ratio, and they are highly correlated with each other. So that is interesting and obviously deserves a test, which I'll go over here in just a little bit. But still, it showed that there are differences here. And then what we did is we said, okay, are the most toxic or metabolite-rich blooms linked to this nitrate to ammonia ratio, at least from the empirical data that we have? And the answer to that appears to be yes. This graph's a little confusing, so let me explain it. On this side of the red line, we have nitrate to ammonia ratios greater than 5. So this is what you're seeing here. This is less than 5. These 2 plots are essentially duplicates of each other, only in different nitrate to ammonia ratio conditions. So here you have microcystin for the amount of nitrogen fixing biovolume. Okay, so this is the metabolite per biovolume on the Y, and these are your groups, microcystin per fixer, per non-fixer, when it's greater than 5. The comparison and why these symbols match up are because that is the different nitrate to ammonia ratio conditions. Hopefully that's not too confusing, but what you need to know is this is on the log scale. So the difference here to here is almost an order of magnitude. They're just simply, and if you go through, you will see this link that in all cases, they were significantly higher when the nitrate to ammonia ratio was less than 5. So when that's less than 5, we found the metabolites are way higher, the concentrations are way higher. So why? Why would this be? Is there a reason again, or is this just correlation? Well, one reason could be that it takes less cellular energy or assimilation time to use these reduced nitrogen forms. They can be taken up passively by the cyanobacteria. But nitrate has to go through a few different steps. So you have to have nitrate, then nitrite, and then ammonia. And all these are active. It takes energy. It takes effort to bring this in. So you don't need reductase if it's already in this form. You don't need nitrate or nitrate reductase. So one reason would be, well, maybe nitrogen just builds up faster, gets assimilated faster. Again, nitrogen-rich metabolite in terms of the toxin. You have more nitrogen. What are you going to do with it? Maybe you put more of this metabolite out because you just have so much nitrogen, especially when the reduced form is there. So what about for these carbon-only metabolites? They don't, geosmin and MIB, there's no nitrogen in them. Why would they be higher? And so one thought is just, well, maybe when you switch from an active way to get nitrogen to a passive way, you are saving energy, and you can reallocate that energy from reducing nitrate to start building more carbon metabolites. Carbon, nitrogen, phosphorus, all these things are tied together within the cell. Or maybe the conditions where reduced N is high compared to when oxidized nitrogen is very high. Metabolite strains, remember there are only certain strains that can produce these metabolites, grow over non-metabolite strains. So maybe it's competition, maybe it's something cellular, we're not really sure. But this sort of summary conclusion from this part, or this empirical study, was that it looked like nitrogen form really had an effect on cyanobacteria, but also the metabolites. That ratio of nitrate to ammonia could be an important structuring mechanism. Maybe it is also driving some community composition changes, especially for the N2 fixers, or the non-N2 fixers, I'm sorry, right? Something is happening there with ammonia and these cyanobacteria that cannot fix nitrogen. Whether it's mechanistic or not, which we're going to test in the next part, it at least could be used as a predictor to say, look, this is when you can expect a lot of these metabolites. So the same question gets asked: causation, only correlation, and we find that out by testing in large-scale mesocosms. So in 2018, at our field station, which I'll show you in just a second, we have large-scale tanks, and we tested this out. So the first thing we did is we filled them with on-site water, and I'll show you pictures of this in just a second. And then we put some of this Milford scum, or at least the goal was to put some of this Milford scum in each one of the tanks so that they had this Milford water. They had an inoculum into all of the tanks. So we could test what would happen on Milford in this case. So we had control tanks. We had 3 of those. We had some where we just added nitrate and we just added ammonium. And if you're just adding a lot of N, again, that increases the nitrogen to phosphorus ratio. And hopefully it ultimately makes P deficient or limited. We also, though, added a lot of phosphorus and then just a little bit of nitrate, a little bit of ammonium, or we just straight added tons of phosphorus. That makes the N to P ratio low. You're adding in a lot of phosphorus, but not nitrogen, or very little. And that makes nitrogen become the limited ingredient, which we saw could be important. So we just have some prelim results. So I'll just sort of wow you with pictures first. So the goal was that we drive to Milford, we get a water pump, and we suck it up. You convince your old advisor to sit on top and make sure that the goo goes in the right place, and you suck it up. And so we did that in 2017. It worked great. So when it became 2018, we decided to do the same thing, only Milford didn't quite cooperate. There was no bloom that year for whatever reason, even though the conditions seemed right. Still, we saw that these harmful species were at least a part of the water. They just didn't dominate it like you see on the background of my slide. So still, we had those species. We had all these taxa from Milford being put in these large-scale tanks. So you go back, you pump it up into your truck, and then you go back, you park slightly uphill. You can see the tank set up here, which I'll go back just a bit. Again, you somehow convince your former advisor to sit there and stir these large tanks so that it's nice and homogenized of all this water that's going in. And then you passively fill sort of a tank that has 140 liters, that has a line right there. So you fill it with this water and then you siphon it into the tanks. And every single tank got this that we worked on. And then you randomize your treatments out there. So here you can see one with ammonia and phosphorus. Here's a control plus nitrate, so on and so forth. There was a secondary experiment that dealt with the purple, but we won't talk about that today. We'll just talk about these ones in red. So here's our trucks, right? This was our little schematic. And then you sample. You start adding nutrients. You add a lot, and you just keep adding those nutrients in those proportions, and you start sampling this water column, which is what this big pipe is here for: nutrients, algae, toxins, all sorts of different parameters. You also, and this is a thanks to EPA Region 7, who was a collaborator on that project, you also can analyze it for the gene for that toxin. We know what it is. And actually, we finally got a shot to analyze all the different toxin forms and types thanks to EPA ORD. You also need a lot of people to run this, right? So Missouri was also involved, the Missouri Limnology Lab and Rebecca North and her team, and they helped out and they gave us these temperature sensors, which take temperature every 10 minutes. And so we got to keep an idea of the temperature throughout that water column the whole time. Then here's our sampling apparatus here. It just has a check valve on the bottom. Put it in, gets the water, and you're able to get an integrated sample of the water column. So we did this, and this is just what it looks like on day 0. Hopefully the pictures do this justice, but you can notice you can see all of these at all 3 depths and all the pictures if you look close enough. If you can't, that has nothing to do with that you couldn't. It's just as the angle of the picture. They're all a little bit different. But again, so you can see the bottom of the tank. That's the white part here in all of these, right? So all our tanks started the same. But after 21 days of gradually adding these nutrients, we added a big initial amendment. And then every week, we kept adding 20% of that, pulsing that system. We saw a very different outcome. Here, you can see the control tank. It looks just like when we started. You can see the bottom. The nitrate and the ammonia, where we only added those, again, that's phosphorus limited or deficient. Well, you maybe can see the bottom-ish. You can kind of see some of those sensors. These were sediment traps we installed. And yeah, you can kind of see those. But the tanks where we added a lot of phosphorus and not very much nitrogen, either none or very little, you can see you cannot see to the bottom. On these, it is very difficult. The ones where we added nitrogen and phosphorus, again, lots of phosphorus, not very much of these different forms, you can see that they develop surface scums. And although this is only one of the 3 experimental tanks, still there was something there, right? These are forming surface scums way more readily than any of the other treatments. So just like you saw on the background of these slides. I want to just present 2 little bits of data that has come out so far. We're still analyzing and working on it. There were a lot of moving parts on this. But the first thing I want to present is this is chlorophyll on the Y. That's in relative fluorescent units. So they're kind of meaningless other than 1 to 2. That's a doubling of chlorophyll. And here's our days from 0 to 21. So you can see we sampled, then we added the nutrients in the Milford water, and we sampled again. So it went up a little bit. There was a lot of chlorophyll in that nutrient-rich water from Milford. But now you can sort of see the treatments, and they kind of go all over. So here's your control in red, your plus ammonia. This is your ammonia and phosphorus, nitrate, and phosphorus, and then just phosphorus. And these colors are the same between these graphs and the graphs on the next slide. So what you can see here is that this little bit of ammonia and a lot of phosphorus and a little bit of nitrate and a lot of phosphorus, they did have higher chlorophyll. Although nitrate also had high chlorophyll, the ammonia only, right? The control had the lowest, but everything else had more. But in terms of who got cyanobacteria, so here this is blue-green algae or phycocyanin, which represents those blue-green algae or the cyanobacteria. What you can see here is that only 3 treatments really grew cyanobacteria. So all 9 of the tanks that we added a lot of phosphorus to, they were the only ones that really came out to have strong cyanobacteria populations. The others did not. There's a distinct grouping here between these 2 treatments. Here you have nitrogen limited, and here you have phosphorus limited. So there is a difference there again, just like we showed in the other experiment. So now we don't have a lot of data on the taxa yet. We haven't gone through that, but I did want to show you the metabolites. So here are all these different forms. These are in nanograms per liter, not micrograms. So just adjust your scale a little bit. So here are 10 different forms. We can sum those all together to get an idea of the total amount of microcystin there. So here you can see that there was a little bit of toxin when we started, and then that kind of just increased. Here's your nitrogen limited treatments, but then by day 21, something happened where this plus ammonia treatment was all of a sudden much higher. Even though we just saw there was not very much cyanobacteria, there was seemingly a lot of toxin relative to these others that had these surface scums. And when we look at all the forms, I want you to key in on just 2 things. One, this LR, I said is the most frequently detected. Well, we detected it in all of our tanks. Every single one, we had a detection. So it is, for whatever reason, one of those frequently detected congeners. But this last day, and in general, this day here in the ammonia only, you can see that some of the tanks, and this RR, that's arginine, that's the most nitrogen-rich form that you can get. And here, another one with this arginine. Not all of them. There are others here that have some nitrogen-rich properties. But these 2 are particularly nitrogen-rich, and that is where we see the detections of that metabolite. So even though cyanobacteria didn't dominate that community, we still see a reasonable amount of that toxin occur in that system. So it does look like nitrogen form has some impact. So to round up this little part and then have a greater summary, just sort of the prelim eyeball and also looking at some of that data was that cyanobacteria dominated in most, if not all, of these low nitrogen to phosphorus ratio tanks where nitrogen was limiting. That is where we saw the cyanobacteria blooms. In the tanks that didn't, there were green algae there. So just like in the experiment that I ran in the early 2010s, greens were not there when we had a lot of nitrogen compared to phosphorus. We're still evaluating and looking at all the taxa data that we got back from algal counts to look at specifically how nitrogen form impacted the community. But it did seem to matter for these metabolites, including this toxin. Again, the most N-rich forms of this toxin were only found where you had this reduced nitrogen form being added in excess, and cyanobacteria were seemingly not even dominant. So again, it provides some evidence, some support that nitrogen form does matter and the proportions may really make a difference. So the overall summary. 1, cyanobacteria blooms have negative consequences. They are not good things. They're not good things for the humans that rely on that water. They're not good things for the ecosystem. They're not good things to live around, and they have detrimental effects. Additionally, resource ratios, the resources that fuel these blooms like nitrogen and phosphorus, can help predict what conditions will increase the probability of a bloom. The nutrient ratios matter. There also seems to be the proportion of non-substitutable like carbon, nitrogen, and phosphorus, and substitutable resources like nitrate, ammonia, organic nitrogen forms. They interact, or at least they seem to drive community composition. And for whatever reason, still yet to be totally elucidated, they somehow are indicative of conditions favorable to their metabolite synthesis or those metabolite producing strains that produce these noxious and toxic compounds. With that, I just want to thank tons of people. I know that I am at 48 minutes, so I'm reaching the end, but just know that there were so many people on all of these projects, the empirical ones and the large scale experimental. I mean, just so many undergraduates, technicians, PIs, you name it. These were big, large scale tests, and they took a lot of people. And I'm so thankful to all the people on this list and all the ones that I've probably missed because so many were needed. And with that, I will leave it to questions, and that's me standing in one of those large scale tanks just to show the size. I'm 6'1", I have pretty long arms, but I'm not even getting close to touching around that tank. So with that, I'm happy to take any questions from the group, or maybe Ben, there are questions that have come in.
Yay, good work. Thanks very much, Ted. So I'm gonna stop the recording now, and if anybody has a question or a comment or even an announcement for the group, just send me a message in the chat, and then Ted can call on you or we'll pass it along, okay? I'm just gonna stop the recording now, and we'll post that.